We are going to look at how sucrose moves from leaves to the roots and from the roots to the leaves. So you will find out that our leaves are going to be the source, and the roots are going to be the sink, like that. So, and at some point you will find out that the roots are the source and the leaves or some other organs that are going to be utilizing this sucrose or the carbohydrate being the sink. So it happens in two directions. If the leaves are the ones that are providing the carbohydrate, we call them the source. Then if the roots are the recipients, we call them sink or the sink. Then if the roots are the ones that are giving in carbohydrates, so supplying carbohydrates to any organ, the organs may be the leaves, the fruits, anything. So if the roots are the ones supplying, these roots are going to be the source. The leaves are the recipients, then we call them sink. So usually students think that leaves always are the source and the roots are the sink. No, it's all about who is receiving and who is donating. So we shall mainly focus on mechanism of translocation. We know translocation is transport of these sugars within the plant. And under this, we shall first look at the process of sugar loading. How is sugar loaded from the leaf or from the mesophyll cells to the phloem. That process there, we have to look at it under sugar loading. Then we have to look at mass flow. These are the theories that we have to study about and look into. We have mass flow, electroosmosis, and cytoplasmic streaming. Those three processes or theories. Then within these theories, we shall focus on the process or how things are happening within that theory. Then we shall also focus on evidence supporting the theory and the weaknesses of the theory. So this recording is focusing on mainly the process of sugar loading. It's what I'm mainly focusing on for today. Then I will make a recording for mass flow and electroosmosis, and our last lesson will be on cytoplasmic streaming. But let us first deal with the process of sugar loading there. So under this, I'm having my illustrations here to guide us, and from this we are saying that we have the sieve tube. So the sieve tube is this terminal there. We have our sieve plates there. So this is the sieve tube. It has safe plates. Then we have cells that are lying within this region. Cells that are lying within this region, we shall call them companion cells. So companion cells are lying within that region. Then these are the sieve tube cells. Then this side is where we are having our source. You may think of the mesophyll cells, as we said, any cell that is carrying out photosynthesis. So that is it. Then we shall go on and identify this molecule here. So this is our hydrogen pump. So that one is our hydrogen pump. And that hydrogen pump is going to be pumping, is going to actively pump hydrogen ions using up ATP. So this one is an active process that requires ATP. So our hydrogen pump is going to be pumping hydrogen ions from our companion cells outside there. So as the hydrogen pump actively pumps hydrogen ions from the companion cells to the outside, what happens is that ATP has to be broken down to ADP, and we harvest that energy. So that's the energy that we are going to use during this movement of hydrogen ions. Then the concentration of hydrogen ions is going to increase outside the companion cells. As the concentration increases, so the concentration increases there, it's going to bring up a gradient in which hydrogen outside is the hydrogen concentration is high, and this side the hydrogen concentration is low. So there is a gradient that is going to be established there. So hydrogen will start diffusing into the companion cells. As hydrogen diffuses into the companion cells like that, it's going to come along with our sucrose. So this one is called co-transport. This one is co-transport. And there are certain proteins that are responsible for that. We shall call them co-transport proteins. We have it here. So as hydrogen moves back into the companion cell, it's going to induce sucrose to also move along the same gradient like that. Now, so as sucrose accumulates within the companion cells here, we have to know that its concentration is increasing within the companion cells. Meanwhile, the concentration of this sucrose in the sieve tube cells or in the sieve tube is relatively low. So if we have a low concentration of sucrose this way and a high concentration within the companion cells, what happens is that this sucrose is going to start moving from regions of high concentration to regions of low concentration. So you end up having sucrose moving from the source to, or from the cells that are manufacturing it, to the sieve tube cells, like this. So as sucrose starts accumulating within the sieve tube cells, it will start flowing down. So this process of moving down till the roots is what we are going to look into next. So this is the process. We shall go through this theory and see. So here we are having an active hydrogen pump driven by ATP, lowers the level of hydrogen ions in the companion cell. This one. How is it lowering the levels of hydrogen ions? Is by pumping it outside. So the hydrogen ions are actively pumped from the companion cells into the apoplast. The apoplast are the spaces within the cells, and this one is utilizing ATP. Then hydrogen ions then flow down a concentration gradient. So we have hydrogen ions flowing down a concentration gradient through carrier proteins into the sieve tube element. So we have these, this is what they are calling carrier proteins. But we've seen that these carrier proteins are co-transport proteins. So as hydrogen accumulates outside, it has to start moving in from regions of its high concentration to regions of its low concentration. So then we have sucrose molecules are transported along with hydrogen in the process known as co-transport. So sucrose molecules are transported along with hydrogen in a process known as co-transport. These carrier proteins are also known as co-transport proteins. So this is it, the process of sugar loading.